Bienvenidos a Sapiens Wisdom. Es un honor presentar hoy al Dr. Randy Schekman, que es biólogo celular y ganador del Premio Nobel de Fisiología o Medicina en 2013 por sus descubrimientos fundamentales sobre el sistema de transporte vesicular en las células, un proceso esencial para la comunicación y el funcionamiento celular. El Dr. Schekman es profesor en la Universidad de California en Berkeley e investigador del Instituto Médico Howard Hughes. Su trabajo no solo ha revolucionado nuestra comprensión de cómo las células organizan y transportan proteínas, sino que también ha tenido un impacto profundo en la biotecnología y en la medicina. Además de su labor científica, ha sido un defensor del acceso abierto en la publicación científica, promoviendo la transparencia y la equidad en la difusión del conocimiento. So it's a true privilege, Dr. Schekman, to have you today. I will make a spotlight for you, for everyone. There you go. Well, I will start maybe with Marvin, who will ask the first question.
Ok, buenas tardes. Mi nombre es Marvin. Un honor estar frente a usted. Leí su biografía y realmente me admiró y me sacó una lágrima. Mi primera pregunta es, de acuerdo a su investigación en su proyecto del descubrimiento, con lo cual ganó el Premio Nobel, ¿usted sabía que era algo trascendental en ese momento o nunca lo pensó en ese
Okay.
momento?
Well, the first question from Marvin. First, he says that he was very inspired by your story. He read your biography and was in tears actually by reading all your work and all your story. And his question is regarding your discovery with which you won the Nobel Prize, and if you were aware from the beginning of your discovery that that this would be so fundamental for science, if you were aware from the start.
Thank you for the question. I remember the moment when I first was able to see the work of my student Peter Novick, who had cut a thin slice through the first mutant that we isolated. And he called me down to the electron microscope room to see this image of cells just chock full of vesicles. I knew at that moment that we had an important discovery and that it would provide us with an opportunity for the next 20 years or more to discover the molecular mechanism of protein secretion. So I knew at that moment that it was big. I had no idea at the time that the discovery would reveal the same mechanism in human cells. That only came later when we were able to clone the genes that we discovered in yeast. And then when the human genome was sequenced, it became clear that the same genes were evolutionarily conserved over 2 billion years. It was not entirely surprising that discovery was made subsequently, but it certainly was something that amplified the importance of our original discovery.
Great. Thank you for your answer. Some of the students can speak English. All of them can understand English. So Marvin understood your question, and those who can speak English will ask their questions in English, and for those who only understand it, I will make the translation. Okay, now we will have Nadia Padilla from Honduras, who will ask the next question.
Good afternoon. My name is Nadia. It is an honor to have you with us. Once again, thank you for your kindness in attending. Your life is an inspiration to many young people and adults in science. If you allow me, I would like to ask you the following question. We read about your history with a microscope. Could you tell us more about that and how it ended up in the Nobel Museum?
Sure. So the microscope was my pride and joy in high school. And but then I went away to university, and I left the microscope at home. And fortunately, my parents saved it for me. And then many years later, when I moved to Berkeley, where I'd been a faculty member, and I had a family, they sent it up to me for safekeeping. And I kept it in my home, but neither of my children showed any interest in science. So the microscope just sat and collected dust. And then when the Nobel Prize was announced, sometime later, every year, the museum sends a notice to Nobel laureates to them send in some, memento, some piece of equipment that reflected somehow their important discovery. And I thought I didn't really have anything in my laboratory here that was of interest, but that microscope was not doing anything. So I dusted it off and sent it to Stockholm. And they have it on display. In fact, just last December, my partner and I were in Stockholm to join the celebration for a former PhD student of mine who won the Nobel Prize in Chemistry last year. And I went over to the Nobel Museum, and they showed me the exhibit of my microscope, together with a little caption in English and in Swedish about how I ran away to the police station to report my parents for taking my money. And that resulted in my getting that microscope. So if I had imagined when I was a kid, 12 or 13 years old, that this microscope would end up where it is, I would have thought I was crazy.
It was worth fighting for.
Yeah, I don't think my father felt that way at the time.
And does it work? Does it still work? The microscope?
Well, I imagine it does, but of course it's just on display. It's not being used for anything.
Yeah.
It's on display along with other artifacts from other laureates.
Yeah, great. Okay. Now, I don't know if Isaac has a question. Okay.
No sé si me podría ayudar a traducirlo, doctor. Pero
claro,
mi pregunta,
sí. I will translate.
Mi pregunta es la siguiente. Estuve leyendo su biografía, muy interesante, y una de mis incógnitas y por las cuales vi es que cuáles fueron las motivaciones y las experiencias que llevaron a él a redirigir su carrera, porque él dijo que de inicio él tenía una perspectiva de estudiar medicina y probablemente patología. ¿Y cuál fue la motivación que lo llevó mejor a enfocarse a lo que es la biología molecular?
Perfect. Isaac said that he also read your biography. It's an incredible biography, and he was particularly amazed by a part of your story in which you change the direction from your interest in medicine towards cellular biology. So he would want to ask you what were the motivations or the experiences that you have that led you to make that decision.
Thank you, Isaac. Before I went to university, I was really just so interested in microbiology. And that was the origin of that story with the microscope. Every year, I did a science fair project, entered into local and state competitions. And so my motivation was because of a passion for biology. But I had no family members who were scientists or educators or physicians. But having grown up in a Jewish family, the idea that their eldest son would become a real doctor was, I'm sure, a source of inspiration and pride for my parents. So naturally, when I went to university, I thought, okay, well, I want to become a physician. But in my first year at UCLA, I took an introductory chemistry class, and the professor was really inspirational, and I did very well in the class. And as a result of my grade in the class, I was assigned to an honors section. in chemistry. And that section was just a small group of students taught by a famous Nobel by the name of Willard Libby, who had invented carbon-14 radio dating. And but the really important part of that class was that every student in that honor section was given an opportunity to select the laboratory of a different chemistry professor to work for that term, a 10-week term. And I chose the laboratory of a young beginning faculty member who was working on bacterial viruses. And so I remember I walked into his office the first day, and he said, well, you know, you don't really know a lot about the work that I'm doing, so I want you to read this book. And so he gave me a copy of what became a very famous book. It was called The Molecular Biology of the Gene. It was the first edition of what became a very important textbook by James Watson, one of the discoverers of the structure of DNA. And the book was remarkable. I read it from cover to cover because it treated the subject of molecular biology and the emerging discipline of understanding genes and chromosomes in a way that was not done in a standard biochemistry textbook. And so this book changed my mind completely. That 10 weeks experiencing working in the laboratory and reading that book convinced me that no, I wasn't going to be a physician. I wanted to be a scientist. So I came home that summer and told my parents that I wasn't going to be a real doctor, and they were disappointed, but they got over it. And then when I came back for my second year at university, I found another laboratory willing to take me on as a research student. And there I learned about bacterial viruses and how they replicate their chromosomes. And that experience really set me on the path that I've taken to this day. So I, of course, have no regrets. I don't think I would have been a very good physician, but I've been a pretty good scientist.
Great, great. Thank you for your answer. The next question will be made by Marvin, who has the next question.
Okay. Siempre en el momento de sus descubrimientos, ¿cuál era su estilo de vida mediante practicaba esa parte?
Okay. Marvin now is interested in. Oh, there you go. Marvin is interested in knowing what was your lifestyle during the years in which you make your discoveries. How was your life and going back then?
My life was very busy. That discovery was made when I had my first child, my son. And so it was busy because my wife had taken off from work to be at home to raise our son. And so I had to do double duty, helping at home and running a lab and getting research grants and writing papers. It was a very challenging time in my scientific career because I had many new responsibilities. I had to teach undergraduate students, and I had to work extra hard to bring in new students to my lab and to initiate the program. I had no experience managing people or teaching undergraduates, so this was all new to me, and I struggled. I wasn't used to teaching undergraduates who weren't as passionate about science as I was. They were smart, but they weren't driven by the science. And so I had to learn to adjust my style so that they could appreciate basic things. And I value that experience. And I since then I teach undergraduates almost exclusively, and I quite enjoy it. I enjoy being able to explain myself to people who are just beginning and to get them enthused about the subject. So those first years were really important to launch my career.
Great, thank you. Now I will make the next question from our group that was suggested from our group, and it says, if you how do you manage adversity? How do you manage the hard times. If you found people during your career that made it difficult for you to advance in science, and how do you overcome those challenges?
Well, of course, not all students or postdoctoral fellows are equally talented or equally dedicated, and so you have to take them at their face value. Some students are fantastic, and they don't need much direction. My experience is the best students know what to do next. They have a result, they know what to do next, and they don't need to or want to be told. And I give those people the freedom to pursue things their way. The students who need help, I ask them to come to see me regularly, and I sit and I talk to them and I give them guidance. And the goal is to get them to achieve as much as they can. These are students who will, have a career in science. They won't necessarily be doing really advanced work, but they will have a meaningful life, and they will enjoy science. So you have to, my experience is to adjust my style, adjust to meet the student at their level.
Okay.
I've seen other styles. My own PhD advisor, who was a brilliant and highly accomplished, another Nobel laureate, had a very different approach. His approach was very demanding. He didn't adjust to other people's personalities. You had to do things his way or else. And he could be very verbally abusive and cruel. He could be cruel to people. And I didn't like that. But that wasn't going to be my style. But what I got out of him was the brilliant way that he approached a scientific problem. And so I valued that experience. But I realized when I started my own lab that I had a different personality, and I had to adjust my approach to what I felt comfortable with.
Yeah.
So there are, of course, some people that I've dealt with who are impossible to deal with. I've had a few people in my life who were just impossible. A small number in my lab, but the major problems I've had have been with colleagues, other professors who are just impossible. And I just try to ignore them if I can.
Did you ever stand up to your professor?
I did, and he didn't take it well. I used to. So this was in the Stanford Department of Biochemistry many years ago, and we had an annual retreat where the whole department came to some conference center, and people had to talk and speak. And I would use those opportunities to make jokes about him. And it always got a good laugh from everyone except him. So we ended on not so good terms. But later on, as my success grew, I think he finally came to appreciate me. But he was a very difficult person, very domineering, stubborn. In some ways, stubborn to his detriment. He had a certain style of doing science that he insisted was the right style, and he wasn't flexible at all. And I think in the end, it did hurt him. But he was, in spite of that, very successful. But his personality, in fact, the way he handled people would not be accepted now. He could not do it in the current environment with being abusive and cruel. He would get into big trouble.
Yeah. Well,
and back then he was so famous that it was simply tolerated.
Wow. Well, maybe there are good things today that are in the system in place, so to prevent those kind of so it's a good thing.
There are good things. That sometimes they're a little bit too heavy-handed, but they're good.
Yeah. You're right. Well, now I believe Nadia has another question.
Thank you. We watched one of your TED talks about publishing important paper in the life science. So my question is the following. What will you change to improve the peer review process for scientific publications?
Why would the what change?
What will you change?
What would you change in
What would I
the
change?
in the peer review process
Yes.
Yeah.
for
Well,
publication?
you may have read some of my opinions on the subject. I've very strong feelings about the problems that we face in publishing our best work. When I was early in my career, very early, and as a student, the most prominent journals were run by scientific society, where active scholars were handled the papers as editors and reviewers. But over time, commercial concerns, principally the major publisher Elsevier and Springer Nature, have become more powerful. And many investigators feel that they can only have a successful career if they publish in those very so-called high-profile journals. And the problem with those journals, there are many problems. One is that they don't use active scholars to handle the papers as editors. They use professional editors who were trained as scientists, but many of them have for decades not been in a laboratory. And so they don't know the modern techniques. They may go to meetings and learn about things, but they are not intimately knowledgeable about exactly what's going on. And yet they are the ones who are charged with making the decisions. And the decisions are often influenced by false measures of scholarship, such as the journal impact factor, a very misleading number, really inappropriately measuring scholarship. It was never intended as such a measure, but it has become a measure of success, and people try to publish in journals that have a high impact factor. In addition, the professional editors, really, they're basically paid, their salaries are they're paid to sell magazines. Whereas the scientific editors of journals that are run by scientific societies are there to promote scholarship. They're not in the business of selling magazines. So the whole system is, I think it's broken, it's corrupt. These commercial publishers, I call them pirates. I think they now have been forced to accept open access of publications, which is a good thing. But they've also realized that they can charge a fortune for the privilege of publishing a paper in a journal like Nature. Just yesterday, I was visiting with a minister of education from Brazil, and I've done so visiting with such ministers in Argentina and in Chile, to talk about the problems that you have in Latin America with affording the open access fees to publish in these so-called prestige journals. It is basically not affordable with your budgets.
Yeah.
And yet these journals make a huge profit. They are among the most profitable businesses in the world. Their profit margin is I think it's obscene. And so we have a real problem. And what I had urged the minister from Brazil to do was to organize not only Brazil, but all of Latin America to force a negotiation with Elsevier and Springer Nature to force them to greatly reduce the profit that they make on papers that are published by Latin American scholars. This is the only way, the power of the purse will be the only way that we can bring these journals under control. And without that, I think they will just continue to make excessive profits. And then, of course, the way the science flows is now indirectly determined by these professional editors. They're looking for the next shiny object, and they aren't interested in deep and enduring scholarship. And so we've handed authority to people whose judgment we would never seek in any other matter. The whole system is corrupt. And I'm afraid there are very few people who speak out like I do about this, and I will continue to do so. But it's I'm not having much traction with my
Yeah.
colleagues who also, they're willing to play this game and fight that battle. It's just really very unfortunate.
Yeah, I totally agree with your position, Dr. Schekman. Actually, well, just to tell you some stories that we of the problems that we have also with open access, which is great for the users, the readers, but for us in Latin America, I've seen groups that are trying to do science, and sometimes they have to sell food to gather some money for months, and then you get the money to be able to afford to publish one paper. So it's prohibitive.
Yeah, some journals will recognize that and will make accommodations for individual scientists to reduce or to eliminate charges. But what you really should do, I think what I try to urge all of my colleagues to do is to refuse to publish in these journals that make an excessive profit. Just don't publish there. Publish in journals that are run by scientific societies where the profit margin is not so important.
Yeah, yeah. Do you think that there's a role that AI could play in this, maybe to evolve preprints into something?
Yeah.
Like,
I think there will eventually be a way around this, but for the time being, the power of these commercial concerns is enormous. Yes, AI can play a role. I mean, I think eventually what will happen is people will post on something like the bioarchive, which is for free, and then the reviews will be done with that version, and decisions will be rendered on that version, and the profit motive will eventually disappear, but not anytime soon.
yeah,
Certainly
it's
not in my lifetime, I'm afraid.
a long way to go.
Yeah.
There has to be some very disruptive event to make that happen.
That's why I urged the minister yesterday to organize not just Brazil, but all of Latin America. If you gathered all the countries of Latin America together and said to these commercial concerns, we will not pay your outrageous fees. And I think it would be okay to boycott these.
Yeah.
I do. I will not publish these journals. And my students know this. They never even raise the subject.
Yeah, great. I actually admire your position, Dr. Schekman, and well, in that regard, I would like to ask you, what is your take on Südhof?
Well, Südhof has served a valuable purpose, but that was before open access started to take hold for most journals. Now that open access is, the commercial journals fought vigorously to try to bring this woman who was running Südhof to tried to get her arrested through Interpol, but she's in Uzbekistan where they couldn't get her. So it was valuable, and it's unfortunate that you basically have to be a pirate to defeat a pirate.
Yeah.
What she was doing was illegal, of course. But I applauded her. But now, as open access has taken hold, Südhof will lose its influence because you don't have to pay now to read the journals. But then somebody pays, and somebody's paying way too much.
Yeah. Okay. Thank you for your perspective. Well, there was Harrison Daniel who wanted to make a question, but unfortunately, he disconnected right now. So
I see him. Is this Harrison Daniel?
Yeah, Harrison. There you go.
Hola, doctor.
Go ahead, Harrison.
Yo tengo una pregunta acerca de lo que es una investigación. Lo que, más que todo, es ¿qué fue lo que le inspiró a usted a investigar lo que es el tráfico de las vesículas dentro de la célula? ¿Qué fue lo que más les dio para poder hacer esa investigación?
Okay. His question, the question that Harrison has is, what inspired you to focus your work on cellular trafficking?
Yeah, so that came about at my transition from being a graduate student to being a postdoc. When I was a graduate student, I worked on the subject of chromosome replication, the biochemistry of chromosome replication, with this my PhD mentor, Arthur Kornberg, who was the world expert in the subject. And it was enormously stimulating, exciting, but very competitive. We had even, more than 50 years ago, it was very competitive. And I was always worried about our competition publishing things before I could. And I didn't enjoy that. And at the time, this was in the mid-1970s, the recombinant DNA revolution was taking hold. And so it was becoming clear that there would be enormous efforts and competition to study DNA, RNA, this kind of thing. And I just didn't enjoy competing with somebody doing the same thing and trying to be a little faster than somebody else. I just didn't enjoy that. So I, through the influence of my best friend, Bill Wickner, who was a postdoc in the Kornberg lab, who had done his graduate work on membranes, I started to read about membranes, and I met some of the important people, such as George Palade, who was later won the Nobel Prize for his discoveries. And I decided to do my postdoc on biological membranes and how they were structured and how they were put together and how they might function. Because I was intrigued by the idea that you could study a membrane using the same tools that had been developed to study nucleic acids. Now, nucleic acids and proteins are covalent structures, but membranes are fluid. They're two-dimensional fluids where things are floating around. So the idea of trying to dissect that more complicated problem of how a membrane got put together appealed to me as a kind of a frontier where there was really, where the modern tools of genetics and molecular biology and biochemistry had really not yet been applied. So when George Palade won the Nobel Prize in 1974, just as I began my postdoctoral work, his brilliant discoveries on the organelles and transitions in the protein secretion pathway were wonderful, but we knew nothing about any mechanistic aspect of that process. No molecules, no proteins, no lipids had been identified to have any specific role in this enormously complex pathway. And so I decided, because of my experiences as a graduate student, that I wanted to study this in a microorganism. But bacteria have a very different process than eukaryotes. And so I decided to work on yeast, Saccharomyces cerevisiae, which had already become a very powerful experimental system to use genetics and later molecular biology to identify essential genes. The work of Leland Hartwell in dissecting the cell division cycle in yeast using genetics was powerfully influential on me, even though it wasn't biochemical at all. Clearly, he was developing a technique that would provide tools with which one could discover molecules. And so I decided, after arriving here at Berkeley, that studying that process in yeast would be potentially a game changer. And I was very fortunate, first in making that choice, and second in getting a spectacular graduate student in my first year, Peter Novick. And he and I then embarked on this process of discovering the genes using genetics. And I was very fortunate. It wasn't all, my intellect. It was just a good fortune and great people and constructive colleagues. So, my PhD mentor thought I was wasting my time. doing genetics. I should have just been doing straight biochemistry. But I thought that was the wrong approach to begin with. And I'm grateful that my colleagues here at Berkeley were more flexible and felt that, genetics was a useful tool. If you really want to study biochemistry, genetics is a useful tool. And so I had a nurturing environment here, and that helped get me going.
Great. Thank you for your answer. I don't know if Nadia or Marvin have the next question. Or
Okay.
right.
Dentro de su juventud, si recorre 30 años o 40 años atrás, mientras estudiaba en la universidad, en pregrado, ¿qué consejos se daría como estudiante? ¿Y qué no haría?
Okay. Well, Marvin has an interesting question. If you could go back to yourself, to meet yourself when you were young and a student, what advice would you give yourself about and the things to do and the things not to do?
Yeah. Well, there were two things in retrospect that I would have done differently. The first is I should have taken chemistry and physics and mathematics more seriously than I did. Since I was at least initially a pre-med, I took the easier courses in physics and math and later in chemistry. And I regret that I didn't study organic chemistry more than I did. It would have been useful. It would have expanded my possibilities, but I'm happy with what I was able to do. That's one thing that I would have changed. Another is I would have tried to convince myself to be a little less judgmental of other people. I think I was insecure in a way when I was growing up. And when I got to school in particular, we're all from more prestigious universities, MIT, Yale, fancy Ivy League schools. And I was just from a public institution. And I felt they probably had some advantage over me in terms of the depth of their education. But that wasn't true. They might have, but they weren't as determined as I was. So I succeeded in spite of that. And I recognized finally, especially when I met my wife, that there was more to life than just working 24 hours a day in the lab. She expanded my horizons. I didn't necessarily want children, but she insisted on it, and I'm grateful to her for that. I love my children, and I wouldn't have had a life without them. So I matured in graduate school and after in ways that it would have been nice if I'd had that perspective when I was younger, but I didn't. But it's too late to change the history. But I am a different person now than I was 50 years ago.
Great. Thank you for your answer. That actually your answer resonates a lot with us in Latin America, because we actually face that same issue, and so when we travel. Yeah.
Well, let me assure you that I know some brilliant Latin American scholars. I have a South American colleague. I think he's from Chile, Carlos Bustamante, an enormously successful scientist. He just gave a very special lecture here called the Faculty Research Lecture this week. And it was brilliant. It was the best of that series of lectures I've ever heard. And so, being from Latin America is not a disadvantage. He obviously had a great education. He must have had very nurturing parents, and he's been enormously successful.
Thank you very much for those words. It's something that gives us hope and energy to continue in our path and to continue to strive to make positive changes in the world and to be part of the global work to humanity, just as you have done. Well, I don't know if Nadia has another question.
Yes, yes.
Okay, go ahead.
What advice will you give to students who are venturing into the world of research? What advice will you give to students who are venturing into the world of research?
Well, I think no matter where you are in university education, I think it's really important to get into a laboratory, to work directly with a professor, and not just to rely on coursework or lab classes, because the act of doing science is entirely different from learning what people have discovered from a textbook. You can't really appreciate what science is all about until you do it yourself, and you can't really know whether you have the temperament to do science at the cutting edge. People who are successful in science are willing to take risks and to be very patient and to keep their eye on the big picture. And people who are easily frustrated and expect everything to work will not succeed in science. You have to be very patient, and you have to remain satisfied by the little tiny victories that occasionally happen, because most of the time things don't work. And you have to be able to understand that and analyze it and try to make improvements the next time you do an experiment. So I have in my lab many undergraduate research students who are mostly intending to go to medical school. In the US, unlike in the rest of the world, you first go to university and then you go to medical school if you choose to, and if you're accepted. And so I have, students who do extremely well on tests. They have the highest marks. They're very smart, and they come into the lab, but they don't necessarily enjoy it. It's kind of routine. It can be boring. It can take very long hours into the night, and then you find out something that didn't work, and you try again, it still doesn't work. I think most students who want to go into medicine decide not to do science because they know that if they're smart and they do well on tests, they'll get into a medical curriculum and they'll work hard and ultimately, if they get a finish and they become physicians, they'll be comfortable and they'll be in an honored profession and doing important things to help humanity. I think it's wonderful, but I think medical students tend to be less risk takers. To be a successful scientist, you have to be a bit of a gambler. And you have to plan your gambling carefully so that you don't lose all the time.
Great. Thank you for your answer. I don't know if there's another question. Go ahead, Isaac.
Ok, me ayuda a traducir. ¿Qué consejo le daría a alguien que tiene la iniciativa de mejorar la investigación y le gustaría así adentrarse al mundo científico haciendo un research fellowship?
Okay. What advice would you have for someone who wants to pursue science and in regard to research fellowships?
Oh. Okay, well, I don't know what fellowships are available to students who wish to remain in South America, but there is an American organization called the Pew Charitable Trust, which sponsors postdoctoral fellowships for Latin American scholars who did their PhD in Latin America, but who wish to come to the US to do postdoctoral work. And it's an enormously successful program. We have a Brazilian scholar here who she may have had a Pew Fellowship for her postdoctoral work at Harvard. And I know a number of Latin American senior scholars who serve on the advisory board for this Pew Trust. I served as an advisor for the program for a few years, and the postdocs they attracted from the U.S. but also from Latin America were wonderful. Were wonderful. Unfortunately, most of them then remain in or they're to build up research institutes that have resources. There are some. I'm aware of one in Santiago that was started by a very famous Chilean scientist named Pablo Valenzuela, who was one of the principals in a very successful biotech company. So he became quite wealthy, and he used maybe much of his wealth to start a new institute in Santiago, Chile. So there can be things like that offer opportunities, but they're very, very few, very few.
Great. Thank you for your answer. I think we have time for one or two more questions. Yeah. Go ahead. Sashenka is in Ecuador.
Buenas tardes. Mi curiosidad, no sé si es que en algún momento tal vez usted dudó de los hallazgos que ya tuvo cuando ya realizó su investigación y tal vez qué consejo le daría a los estudiantes que están atravesando quizá por ese proceso.
¿El proceso de, perdón?
Si en algún momento dudó de sus hallazgos y
okay.
tal vez es que, si le puede dar un consejo a algún estudiante o doctor que estuviera atravesando por esto.
Perfecto, muchas gracias, Sashenka. Well, Sashenka wants to know if you ever had doubt on your results or on your findings, and what advice would you give to someone who may be experiencing that with their results in an experiment or their project?
Yeah. Well, I had doubts, of course, and there were times when I had to back away from a particular project that was just not working. You do occasionally have to make corrections if you've made a mistake. Perhaps my biggest disappointment came when I started as a faculty member, and I had, since I'm willing to take risks, I decided to do something completely different for my own independent research. I had no experience with genetics or working with yeast or studying protein secretion, no experience. But I was full of ideas, and I had been successful in the past. So I wrote a grant to the National Institutes of Health full of my ideas, but I had no preliminary results because I'd only just started. So that grant was rejected, resoundingly rejected. And, with comments that I had no experience, I didn't know what I was talking about. And that was quite a crushing blow because I'd never had that experience before. But I was successful in getting money from other sources, a little bit. And I was determined. I really was determined. I knew what I was doing would work eventually. And then within a year, it just started to work because of this wonderful student, Peter Nobel. So you have to have confidence. And here's something that's really important. If during the course of your graduate work or your postdoctoral work, if you taste success, if you taste the experience of overcoming difficulties and tackling a problem and getting good answers, that success builds confidence. It's really important to build confidence and to have it based on something real, not just your own ego. But you have to taste a little success, and once you do, you get confidence that you can do the next thing that may be equally challenging. And I had, as particularly as a graduate student, I'd been very successful, and that, in spite of now choosing to do something different, I had that confidence of my experience to build on. So you have to find a situation where you can gain that confidence, because if you don't have it, it can be very difficult
There you go. I had some issues with the connection, but okay. So now I will make I will play the audio from a 5-year-old from Santiago de Chile. His name is Santiago, also, and his question is the following.
¿Por qué las células nunca dejan de trabajar?
And that is why do cells never stop working?
Well, they can, but they don't always continue to work. Sometimes they die, and the disease is as a result very often of cells dying. Something else that I'm very involved in is an international effort on Parkinson's disease, where specialized cells in the brain that make one of the chemical neurotransmitters, dopamine, die. They die slowly over many years. They die. And what we desperately need to do is to figure out why they're dying and try to keep them alive. They may not be dividing, but they still live and function. But when they die, that's when bad things happen. Neurodegenerative disease is all about brain cells dying inappropriately. So we need to know how cells can keep themselves healthy and how to prevent them from dying when that death leads to disease. This is the challenge of life science, and it will continue to be a challenge for many, many years. We know so little about the brain. You'll be amazed at the discoveries that come in the years ahead, understanding how the human brain works. It's much more impressive than AI. With AI, which may eventually become more powerful than the human brain, the only reason it works so well now is it requires massive amounts of energy. I mean, they're talking about using nuclear power plants to provide the energy needed for the massive computation of AI. And yet, what we do with the energy of a 20-watt light bulb is all contained within our skull. That's the amazing thing. Much more amazing than AI.
Yeah, that's amazing. Yeah. Thank you very, very much for your answer. I haven't seen that perspective from the energy, the comparison of energy from the biological perspective. Yeah, so thank you very much, Dr. Schekman. Well, with this we reach the end of this discussion, and we would like to thank you deeply for the time you're giving us to share your knowledge and to inspire us. I can assure you that all of us that are here and all of the students and researchers in Latin America that are going to listen to this talk will be very inspired to keep on striving and to impact the world, looking up to you as an example of how to do things in life. So thank you very much. It was an honor to speak with you, Dr. Schekman.
Muchas gracias.